We start this afternoon's discussion with a discussion by Ron Farkas from the Food and Drug Administration, the Division of Neurology, CDER, which is the Center for Drugs, and the Division of Neurology. I first met Ron a long time ago, many years, when we went to the FDA. I think it's been 5 years now. We talked about this unmet need called Duchenne muscular dystrophy. We met with the neurology division and they listened to our stories and they listened to this need. And that was followed by a question and a discussion about the equation of benefit-risk. Because we said in Duchenne that risk-benefit equation is different than it is perhaps in other diseases. So that followed with the pilot benefit-risk study that we did and provided to the FDA. And in a recent meeting, Dr. Billy Dunn said, we get that equation and we get it very well. As you know, they released guidance on June the 9th. And one other thing that I can say about Dr. Farkas, I can say lots of really good things, but one in particular is last year we developed a relationship, public-private partnership with the National Institute of Heart, Lung, and Blood. The reason for the partnership, as you might imagine, is because we have the question in Duchenne muscular dystrophy, what do we need to know about this heart? How well is it characterized? If you extend function for years and years and years, does that negatively impact the heart? What is this heart disease that occurs in Duchenne? So we had a meeting with HLBI. You know, in Washington, everything's an acronym. So we met with HLBI. There were two working group chairs. One is Beth McNally out of Northwestern in Chicago, and the other's here in the audience, Larry Markham out of Vanderbilt. So that's the pediatric cardiologist and an adult cardiologist that chair the working groups that have now divided into 3 sections. However, we had a day-and-a-half meeting to think about this. What do we know? What are the questions we need to ask? And what do we need to know in order to better understand and treat or prevent heart disease in Duchenne muscular dystrophy? Dr. Farkas came and spent the day with us because he wanted to learn all he could about the heart of Duchenne. So he really has been a friend to our community. He gets it, which is really nice. And so today I present you Dr. Ron Farkas from the FDA.
Oh, thanks, Pat. I really appreciate that introduction. Make sure I know. Well, before it, I'll probably.
Stephanie?
Oh, there it went. Sorry. So I actually wanted to spend kind of a second more on this first disclaimer slide than perhaps people often do. And I think that one piece of reassurance that I want to give everybody in the Duchenne community and actually everybody who watches the FDA is that while all the opinions of all the scientists on a team are valued, there is an incredible spirit of cooperation among everybody at the FDA. There's incredibly clear communication. Dr. Woodcock doesn't know all the details that the people in the division knows, but we talk to her frequently and in detail and answer all of her questions. And that actually goes up to the to the commissioner, which we don't have it right at the moment, but acting commissioner, and to the Department of Health and Human Services and on up through the executive branch. And I think, too, that there is really a tremendous amount of agreement among all the people working on these drugs, and there's a lot of gray areas, and sometimes decisions have to be made about what to do, and then when it's very gray, it's hard to know. But I think that you can be assured that the FDA is a very internally open organization, considering everybody's opinion within the organization and from without the organization. This first statement on the slide, I'm glad that Pat said that she thought that we understood the urgency of the situation. But I think that first bullet point illustrates a little bit of the difficulty of communicating clearly what the FDA is doing and why. And I was looking at this before the talk, and the last phrase of that first bullet point, the urgency to make new treatments available. And I was thinking about what that meant and the exact word choice, and I think that it could be misunderstood. What the FDA does is try to determine if drugs, or what it first tries to do is determine if drugs are effective. And that's what takes all the time. That's what takes all the time in development. That's what takes all the time when the FDA is taking a look at the evidence. And so perhaps things like this, statements like this, could be misunderstood, that the FDA has already concluded, or that the FDA thinks there's already enough evidence to clearly conclude that the drug is effective, and yet there's some kind of bureaucratic delay or some kind of red tape or something like that. And I also want to reassure everybody that's entirely not the case. So we were in discussions, for a long time, of course, with the community about the way to approach Duchenne muscular dystrophy. And the FDA issued a draft guidance for industry to help drug developers understand FDA thinking. And this was the first time that a community organization, an advocacy organization, had first gone through great effort to assemble a great deal of information for the FDA to consider when writing a guidance. And we very much value that effort. I think I also want to reassure you that we very much read all the literature. We very much talk to all the experts to try to understand what's going on. But just as an FDA guidance is valuable because it really tries to create a clear message at a certain point in time, we very much value all the work that went into very kind of concisely putting all the information together for the FDA to consider. So actually a lot of the names for these guidances get a little bit confusing. There's the PPMD guidance, which was a draft guidance, and there's the FDA guidance, which we put out, but that's a draft guidance too, and I'll explain a little bit more about that later. But I think the overall message is that this is a scientific endeavor, and that ideas are constantly being updated based on the evidence, and there's nothing static about it. And of course, there are differences between the FDA guidance and the PPMD guidance. But the FDA very much views what's in the PPMD guidance as is incredibly valuable for us, for the community, for drug developers. We think that's a kind of an ideal situation. The text on this slide is, I think, verbatim from the introduction to all of our guidance documents, and it explains what their purpose is. And I should stress that the guidance documents, even though a huge amount of work goes into them and many people read them and they're vetted and finalized and published, that represents current thinking, and current thinking can change at any time. There's nothing static about that. It's not binding, and that I'll mention a little bit later. Not only is it not binding, but we try to be careful not to limit innovation. And I think in some sense, what I'd like you to understand is that sometimes when we don't say anything, it's because we don't know. And if we say something that we don't know, that could hinder progress. And again, just this idea about alternative approaches. I mean, there's alternative approaches, and also I think that the community should know, and I think this is something that drug developers know, is that if you say something to the FDA and the FDA says, well, we're not really so sure about that. We're not really convinced that you can come back with more evidence and try to explain it again. If you know what you're talking about and we're not getting it, try to explain it again. And that always holds. So then getting back to the draft guidance. So the FDA Duchenne guidance is a draft guidance, and that's open for public comment. And we absolutely appreciate those comments. I mean, those comments can be so valuable. If you read something in that guidance that seems like, boy, that doesn't make any sense. Why are they asking for that? Well, maybe we wrongly asked for something. Maybe we didn't word things right. But we need to hear about how to improve our ability to communicate through these guidance documents. And the easiest way to do that is online. There's something called a docket, and you can submit your comments to that docket. And we absolutely read all of those comments and carefully consider. So then, after some period of time, a final guidance will be issued. I mean, I should say that I'm not exactly sure how long will take to issue a final guidance. I would say that is neither a mark of something going right nor a mark of something going wrong. I mean, there's certainly it's kind of the intention of FDA to issue final guidance, but I think we'll wait to make sure that we know that there's some, kind of enough clarity in the document before we would make it a final guidance. And again, the final guidance is only final in that until the next revision comes out. So I think one key thing that perhaps the FDA hasn't communicated well enough to patients and advocates, maybe not even to drug companies, is that, well, now there's a guidance for Duchenne muscular dystrophy, but we have many guidances that absolutely apply to muscular dystrophy. But they're general, so they're about study design, they're about controls, they're about many things that people often don't think about. They're about studies in animals before drugs are given to people. They're about drug-drug interactions. And so those are incredibly important documents. And so we try not to repeat information in those general guidances in what we would call a disease-specific guidance. So, for example, choice of control group, that's obviously very important to people. The Duchenne guidance does go into a little bit of detail about that and mentions some aspects that are unique to Duchenne muscular dystrophy. The guidance says steroid use needs to be considered. I think that getting into historical controls, one thing to keep in mind with Duchenne is that supportive care has been very beneficial. And I'm not sure if that's stressed in the Duchenne guidance, but it's something that needs to be taken account of when comparing patients today to patients even in the relatively recent past. And that's not saying anything's impossible, and I think that what we try to stress is that most things are not impossible, but there is a necessity for considering how data might not be clear or how it can be reliably interpreted. And I think I could recommend for anybody who is interested or had more questions about historical controls to read this other guidance called Choice of Control Group and Related Issues in Clinical Trials. Because that does go into a fairly exhaustive discussion of the benefits and pitfalls of different kinds of controls. And it applies, of course, to all different kinds of diseases, and that's why it's a standalone guidance. There's other guidances, too, that talk about study designs that might be more efficient, or as efficient as possible. And again, that's not discussed in the Duchenne guidance, but it's certainly what we think about all the time. So this first guidance listed here, adaptive design. This is the idea of, can we learn about how to study a drug better as the study is already ongoing, basically. Enrichment strategies, our guidance tries to kind of explain both kinds of strategies for trying to find efficacy in as quickly as possible and sometimes in studies as small as possible. One way that we think you can find treatments as quickly as possible is to expand enrollment criteria. Of course, many people want to be enrolled in clinical trials who might not qualify, and there can be great advantages to enrolling more people. More people is more statistical power. And on the other hand, sometimes it's quite obvious, this idea of enrichment is that you enroll patients who are most likely to respond to a drug. And sometimes it's obvious, if it's based on a mutation, that, well, of course, these are the only patients that you would enroll. These are the only patients who could possibly respond to the drug's mechanism. But sometimes it's a lot less obvious. It would have to do with if you think a drug is going to slow progression, you would enroll people who were progressing at a certain rate, who were a homogeneous group, and that would allow you to compare your groups in a way that would increase statistical power. So some of the most important things that are said actually about what the FDA does and why, they're said in the regulations themselves. And that really shouldn't be overlooked. And I've reproduced a lot of text here from what we call subpart E. And I won't read through the whole thing, but I think it was, again, some of what Pat was saying about something about flexibility, but something about understanding risks and benefits, understanding the condition, and in a lot of ways, this is a golden rule for us, and we don't ever forget that. And that, again, it's not something that we necessarily repeat in guidance, but it's something that we're always thinking about. And the regulations themselves talk about what we would call adequate and well-controlled trials, the kind of efficacy data that the FDA is looking for on which to base drug approval. And the regulations themselves explain some of the benefits and pitfalls of different designs and list things like placebo-controlled trials, dose comparison trials, no treatment, concurrent control, active treatment, and historical control. So all of those designs are in the regulations. And I think that, to me, they all deserve, and I think to everybody else at the FDA deserve more attention. And it's not just one type, but one of the very informative things I think about Duchenne is what's occurred with corticosteroids over the years. The way that they were first found to be effective, the kind of studies that were used to find that, the size, the duration. And we don't expect at the FDA for other drugs to act like corticosteroids. We don't have any comparative efficacy in mind at all. So I just, I will totally deny that a drug needed to compare in any way to what corticosteroids do. But it is really tremendously useful, nevertheless, to see what scientists are doing, looking at things like the dose response of corticosteroids. And there is this, to me, I'm very fond of what you might call pharmacokinetics and pharmacodynamics. And there really is this incredible power in, for example, comparing more frequent to less frequent doses of corticosteroids to find out, and here's the important thing, not just to find out that, well, maybe high doses work better than lower doses, but the thing is, if a high dose beats a low dose, you know that high dose also beats placebo. A few assumptions have to be made, but those are usually possible to make. So everybody's very interested in ways to not have to have placebo, and that shouldn't be dismissed. And one of the issues is practicality, feasibility. But I think that's always something that can be talked about. I mean, that all involves people and time and money, expectations, when are we going to get the answer? So I'm not saying it's a perfect solution, but it deserves conversation. And so does, will active treatment control. I mean, what that's really saying is comparing two drugs to each other that might work. And there's limitations to exactly what information you learn from a study like that. If both drugs are about the same, if people on both drugs do about the same, you're not sure if both drugs are effective or if neither drug is effective. But if one drug is more effective than the other drug, then you know that drug works, that drug that was better. You don't know that the other drug didn't work, but you know that the drug that was more effective, that it worked. And these are very well understood, controlled trial designs that deserve, I think, more thought. So getting back to the guidance, and I had mentioned before that the FDA tries not to be too prescriptive in what is included in guidances. Of course, it says this is just advice. You're welcome to do a different approach if you want. But still, I think that it's, perhaps it's human nature. There's one guidance I won't mention where an endpoint was put in parentheses. It was, you should use an endpoint like this, or we recommend an endpoint like this type, for example. And it really didn't take very long for that to kind of, through kind of a game of telephone effect, that was a preferred endpoint. And it was really no such thing. So we try to avoid the possibility also of saying things that would be interpreted as us setting limits. So regarding biomarkers, the guidance says that, I think this is verbatim from the guidance, that the FDA has particular interest in dystrophin as a potential surrogate endpoint to support accelerated approval. And then the guidance describes a little bit more about how the amount, the timing, the location, the functionality matter. I think that one of the true things about science, and that all good scientists need to keep in mind, is that evidence is cumulative. And I personally think that one of the impossibilities of talking about kind of in the abstract of what's acceptable and what's not acceptable is that the context always matters. The other evidence always matters. And in truth, in any kind of science, one thing that's often not talked about is that the likelihood that the drug works even before you started clinical trials, that's really important too when you're interpreting data, because you're always trying to figure out, did something occur by chance or because the drug worked? And if I could just kind of digress for half a second and explain what happens when you just go to the doctor for an ordinary test. So an ordinary clinical test is useful for diagnosing, say, the flu, if you have the right symptoms. But there's false positives. That test, 1 out of 1,000 times or 1 out of 100 times, is going to give a positive result, even if you don't have the flu. Or maybe a better example is a pregnancy test. I'm pretty sure that if you gave a man a pregnancy test, or 1,000 men or 1,000,000 men, you'd get back some positive results. But the interpretation of that positive result really depends on the likelihood that that condition actually exists. And so the FDA is keeping all these things in mind and synthesizing different kinds of data, positive evidence, negative evidence. Well, that's science, but it's, you know, it's the art of science. I mean, there's art in science just like there is in medicine. Art is maybe another word for gray areas or areas where a lot of judgment is required, and areas where you're not sure. So the guidance also mentions MRI as a method that I think the thing that I would like to stress about this is it's a method that looks at the tissue level. And I think one productive way to view biomarkers is kind of different levels of activity. So in development, a scientist developing a drug would look to see, well, does the drug get to the tissue? Does it bind to the receptor it's intended to bind to? Does it cause the cellular change, that first cellular change it's supposed to change? Is there something downstream, still at the cellular level maybe, that changes? What about at the tissue level? And then on and on, up at each different level. And so one thing that's attractive about some of the MRI measurements is that it could kind of go together with changes that you expect to see and maybe you do see from a drug. And that really helps strengthen evidence that something's doing what you think it's doing. And just as a very general statement, we didn't in the guidance list all the different possible biomarkers, but we listed the kind of the criteria, tried to explain that a little bit more. Something that I was just explaining, biochemical, cellular, tissue level, those can all be very useful biomarkers. And it all does come down to something that's very hard to describe before you see the data. It's how much scientific data is supporting this, and also how reliable is the measurement. Just like in clinical trials, reliability really does matter. So this is just talking a little bit about some of the things that the guidance really distinctly doesn't have. And kind of as a general concept, the FDA is talking about drug development, talks to people about drug development, and not about clinical care. The practice of medicine, I think, is what we would normally call it. And so that information is incredibly valuable to the FDA for us to learn about how to make recommendations to sponsors, about how to run studies that will work, about how to select endpoints that are likely to be the most efficient. And we like to hear that information from other people and to get that knowledge, but it's something that we wouldn't ordinarily include in a guidance. Again, not because we don't value it, but because it's something that other people are teaching us, and we're not teaching other people. So that's all that I had to say, and I'm happy to take any questions if I didn't talk too long.
So we're open for questions. Any question you might have, and please refrain from asking a particular question about a specific intervention or drug that's being developed. Questions?
My question has to do with the design of the studies, and you'd mentioned the need for a kind of a homogeneous population, and we've been struggling in the community trying to make sure that we're as inclusive as possible into these studies, whether the studies require ambulatory versus non-ambulatory or steroids versus non-steroids. And I'm just wondering if you foresee in the future, as we get maybe more sophisticated with our biomarkers and our endpoints, if you see where we'd be able to be more inclusive with who can participate in these studies.
Yeah, well, I mean, I think that there's I suppose, two different strategies. I'm not quite sure. I'm going to try to come up with an analogy. I'm not quite sure if it's the right one, but so in there's some animals that have like a million young, and some that have one, and these are just different strategies for trying to get to the same goal, and I'm sure that's a terrible analogy, but so again, try to forget it if it was wrong. But I think with clinical trials, it's kind of the same thing, I hope, and that is that if there's a reason to think that a drug will only work in a subset of patients, it's really good strategy to limit enrollment to those patients. It really ought to be a good reason, though, that you're limiting enrollment to those patients. It doesn't have to be proven, though. A good theory can be a reason to do that. And again, the genetics is one example. The issue of wider enrollment, I think, too, evidence is additive, and I guess that sometimes it is good to use an endpoint focused very much on the functional abilities at a certain disease stage. And if they're different across different patients, that adds some complexity to the study. Complexity can be worrisome. Kind of in concept, though, you can combine data from different endpoints. There's no problem with that, the endpoint that's right for the patient. So anyway, I hope it wasn't too meandering there, but I guess the answer is that there are good ways with either strategy.
Other questions? Thoughts?
I just had a question about the control groups. If you look at two active drugs, you certainly can have both of them be inactive. And one could be doing harm and one could be doing nothing. But I'm more interested in understanding a little bit more your thoughts on looking at two levels of dosing of the same drug and how you would use that.
Right. Well, doing a dose-response study is a very well-understood way of doing studies for drugs. No doubt it increases the need for more patients. That's clear. But this can be calculated, basically. And so there's a trade-off. I think that with all these different study designs, I think I can be confident to say that if speed is the priority, that a placebo-controlled trial is always faster. And I think depending on how you might model it, I think the FDA would pretty much always say it's going to be advantageous, probably even down to individual patients, that they would get a drug that was effective faster with a placebo-controlled trial. And it all depends on the exact numbers that you put in. So then looking at a dose-response trial, we understand that time matters. Time matters in drug development. The FDA has made sure it's well educated on the value in terms of drug development of 6 months or a year or whatnot, or enrolling patients. And from some of the things that have been seen recently, 6 months earlier to marketing can mean hundreds of millions of dollars of difference to a drug company. I don't say that that's a small issue at all, but from a scientific perspective, if it is a priority, which is a completely reasonable priority, to diminish use of a placebo, then a dose-response study can show that the drug is effective.
Good afternoon. I'm
Right.
trying to understand something, and I'll try to be brief. So Timmy's stuck in the well, right? You know, and the parents, you know, it's a situation, right? Clearly understand. Is intellectually, I'm trying to deal with. I understand we need effective drugs, but is there a possibility that we're being so careful, so methodical, so diligent that we're actually losing more boys in the process? Now, I know that's not a new concept, but, you know, I struggle as a parent. I'm not asking for false hope. I certainly don't need the FDA to show me, you know, some snake oil. I get it, you know. But I really intellectually and emotionally struggle with, you know, hope. Hope is valuable too. And I'll try to wrap up my question. Does the FDA kind of get the urgency? If it's safe, can we risk the efficacy? And I know there's a lot of money at stake, but we're spending money on everything, so I'll stop there. Thank you.
Sure. Well, I think there's two answers to that question. And I totally appreciate that question, and I don't think the FDA talks enough about that. It's kind of an uncomfortable topic, but you definitely need to know where we're coming from. And I think the first answer is that this is totally the honest answer, and that is that we are not allowed in our jobs to say that a drug can be used if we don't think it's either effective or under Subpart H reasonably likely to be effective. We simply can't do that because of the laws. The second part of that is that I think people at the FDA believe that's the best thing to do. That's in large part why they work there. I don't think there's a right and wrong about that. I mean, I think, again, it's possible to envision different scenarios, and I'll posit to you that this is the big factor that matters. What matters is the likelihood that these drugs coming out of the animal studies or the laboratory studies, the probability that those drugs are actually effective. Everything that we do in science depends on that. So if the likelihood was 50% even that the drug coming out of the animal study was effective, then it wouldn't make sense what we're doing. You say, well, you know, it's 50% chance it's going to work. Let's just use these drugs and figure it out later. I mean, let's take 2, and then it's going to increase our chance even more. And you know, people are incredibly optimistic. People who develop drugs for a living have to be incredibly optimistic because the batting average hasn't been 500 throughout the past. So what's happened in the past is that the likelihood that the drug works when everything is pointing in the right direction is a lot lower than 50%. And so so then not knowing if a drug works will probably, I mean, the odds are against you if you don't have enough information about that drug. So science is always improving, and maybe we're better now than we were in the past at finding drugs that are going to work. Even before we test them in patients, we have much higher confidence that they're going to work. And the FDA is completely open to that possibility and looks at the data as it unfolds. Looks at the data not just in Duchenne, but from every other condition, from similar drugs, from not so similar drugs. So if I may kind of continue, that's half the answer. And I think the other half of the answer is one just about scientific standards and how high should they be. And And I think that that the FDA, I mean, first, if I could explain that the standard for approval of a drug is that it's, or the first part of it, the efficacy standard is that there's something happening that's different than nothing, which that's a reasonable standard, but it's not exactly what patients normally think about in healthcare. They're usually looking for something better than just the barest detectable, better than nothing. And I think too that the way we design studies, they are incredibly powerful at detecting efficacy, and I think that people sometimes worry about that, but we see positive studies with extremely small numbers of patients. There was an approval for a drug for a rare disease that affects blind patients and patients who are nearly blind, non-24-hour sleep-wake disorder, and one of the studies had 10 patients in it, or sorry, maybe it was 20, but 10 patients on drug and 10 on placebo. And we absolutely recognize that that can be enough to show efficacy. And so anyway, just so I can stop talking and take another question, I think too that we're open to discussion. We are completely open to discussion about what the standard should be. I mean, how much evidence is enough evidence? That's something great to have a discussion about. We talk about some of these things. We talk about a lot of these things in our guidance documents, but we're always willing to talk more about it.
So I have two questions. The first question is, for rare disease drugs approved based on a surrogate endpoint, does the FDA require a greater level of safety assurance, understanding that the agency looks at the totality and the benefit risk profile when approving a drug?
Yeah, I mean, I think I understand the question that you're asking, which I've been thinking about a lot, and I'm not sure there is a single answer to that question. The FDA, there's been a great deal of attention now paid to the risk-benefit analysis, to explaining in a diagram, almost like a scale, not quite, that's not quite right, but putting all the main benefits on one side and all the main risks on the other side, and trying to figure out, trying to communicate exactly clearly how a decision is made. And so then, if a drug is approved that's reasonably likely to be effective, well, how much does that weigh on the scale? And I guess the answer would be that you determine how much it weighs on the scale the same way that you determined that it was reasonably likely to be effective, and that is, it's kind of equal parts picking the observations that you think are important and true, and weighing them and coming up with a kind of a formula that seems to make sense, and it'll differ between different scientists. It's expected to, just like everything else in life. Very few things are black and white.
So my second question, and thank you for answering the first one. My second question is, with FDA's initiative on patient-focused drug development, what is the agency doing within the space of DMD in terms of understanding the patient's perspective in their treatment, understanding how they feel about benefit risk? Right.
Well, I mean, I think that we're very receptive to hearing about benefit risk. I mean, we've been interacting with the community, and we're very interested in more formal ways to look at benefit risk. I mean, I think that, I should step back a moment and say, I mean, we value hearing about what people think, but we're completely willing to approve a drug that half the people would decide not to use because they don't think it has the right benefit risk for themselves. I guess at some point, if I'm not quite sure, if enough people in the community thought, no, this isn't safe, then I think at some point we would say, or safe for the benefit, then I think at some point we would have to say it doesn't have an acceptable risk-benefit profile. But we are, the real hurdle is showing efficacy greater than no treatment. And after that, everything is entirely open to discussion.
And just to be clear, we are starting on a phase 2 benefit risk that's going to look at benefit risk across the trajectory of Duchenne to see if it changes at any point in time across the spectrum of the illness, as well as asking that question not only of caregivers or parents, but asking the question of adolescents and young adults to see if they agree or disagree.
I mean, I should just add, I mean, that information is valuable in writing things like labeling. I mean, I don't want to underestimate the value of risk-benefit and the way people perceive things or anything like that. I mean, it can be an incredible burden if we write in the label that a drug, you know, requires a certain amount of safety testing or a certain kind of distribution or tests. I mean, we have drugs like that. We have drugs that cause blindness, and we really work hard to try to figure out what a sensible risk mitigation strategy is. So all this information is incredibly useful.
Thank you.
I have a question and kind of a comment on the process that you guys are using. Is that you guys seem to be looking for some ideal, perfect sample that really doesn't exist anywhere in any science. I mean, we do all the science in the world, physics, chemistry, all these things based on, first of all, not knowing what's going to happen. And when you try it, and then normal distribution and these kind of things don't exist as much as we'd like to think in the real world anyway. So I think we're obsessing too much with finding the ideal sample that really won't exist anywhere. And I think there needs to be a better way to look at it so that we can, include more people. We simultaneously want to include a big group and say we have enough people, but we're also limiting by saying you have to meet this and this condition. So it becomes very hard to do that. And I mean, I've also just studied a lot of statistics. I know in real life, yeah, we want that ideal distribution, but it really doesn't exist. It's just an abstract concept to help us understand things. So I think, how do we get around that issue where you're simultaneously saying we need a homogeneous set with all these conditions, but then we want a big set because it's not big enough to mean anything?
Yeah, well, I think that, I want to be perfectly clear about what the FDA is saying to drug developers. We spend a lot of effort trying to direct people towards designs and studies that are most likely to work. But I'm not sure that that's the same thing as demanding anything. We're consultants. I think that that's maybe something that people don't quite realize, is that we spend most of our time as consultants, and drug developers come to us asking for our best advice, and we give that. And we tell them all the time that if they can find a better way, we are completely open to that. So I personally think that a lot of what you're saying boils down to the chance that you have some evidence supporting evidence. When is that enough? When is that enough? And with one study, there's always going to be a fair chance, whatever fair means, that the drug doesn't actually work. It depends all on... So that's something, we have a guidance, and it describes, well, what would you like to see in a single study? If you just did it once, how much evidence do you think you can get from that study? And so I think we think about that all the time. And kind of, one study is the minimum unit of evidence, even if it's one biomarker study. So and in a way, we're kind of at half a study too, because half a study is really rounds to one study too. So we're looking at half studies too. So I think it's a great scientific discussion to have. I think we can have that absolutely. You know, it does come back to things like you know, the typical way science is done. The typical way science has been done. I mean, all the progress that we've made is actually saying, you know, that looks so promising. Do it again. That's not. I mean, that is in some ways the very basis of the scientific method, and so we're not necessarily saying that. So I suppose that to us and our kind of scientific hats, we're already into a region where there's a high level of uncertainty if a positive finding is really positive.
I mean, it's just like when Edison invented the light bulb, he tried thousands of combinations before he got it to work, and he never knew which one would. So I think, I mean, we always never know what the outcome will be. And I think as a community, we are altogether kind of risk takers and willing to see just in case it might help us. So I think maybe that's part of the thing, that maybe it's not clear, I don't know. But the risk tolerance is also higher here, too. So I think that should account for something.
Yeah, I mean, it's absolutely good to hear what you're saying. I mean, I would just try to reassure you that that we are hearing what you're saying and that we are considering that. We do think it's completely valid.
Other questions? Here's one.
Hi. I think that I might be asking the same question just in a different way, but we saw a slide about how you're opening or being more flexible with your risk-benefit. But then we also hear on the other side the idea of having scientific standards. I think what I would like to be able to walk away with, if it's possible, is how you are going to reconcile those two things. For us, where we're not reading about all the trouble that some companies are having with the FDA, where we're not, like as the other gentleman was saying earlier, I think as a parent we're very, very frustrated. And is there anything more specific you can give us with how you're going to manage those two things, being more flexible and then also like allowing efficacy to overrule the possibility of walking away with something helpful?
Well, I think that one of the main problems is that we have to keep so many secrets at the FDA. I can explain to you in the abstract, in detail, what we're doing, what we would do, what our standards are, and I think those are good discussions to have. And when it gets down to specific things that are happening, because of the way drugs are developed, because of intellectual property, we can't really say very much. I think that one thing that I kind of really want everybody to do who's an advocate or a patient or a parent is to question what they're hearing from the FDA and to question what they're hearing from everybody else. And we are going to tell you everything that we can, and we're going to listen to you about what you're saying. I guess that perhaps in the future there's a way that society could run drug development so that there would be more transparency, and I think some of these issues wouldn't come up. But I think in the meantime, hopefully it does help. I mean, I could explain about the reproducibility, the reliability of Western blot, and we did have a meeting about that. And I'd be happy to take questions about that. I mean, I think that, too, I could maybe translate for some of the things that are said. I mean, I think the bottom line is that we need what has historically been considered the barest minimum amount of evidence to believe that something is ultimately reasonably likely to be true. I want to give you my reassurance about that it's the barest amount of evidence to be reasonably, that word is always there, reasonably assured that something is true.
Hi, I'm from England. Recently, about 2 years ago, the EMA produced draft guidance to which we made submissions, and we recently held a workshop at the EMA. I'm just wondering what efforts there were to harmonise with the FDA guidance and the EMA guidance.
Well, I'm familiar with the EMA regulations, certainly not an expert with that. But it's really important to understand that there are differences, really profound differences, in the regulations for EMA and for the FDA. So in that we have a lot of harmonization. It's called harmonization. A lot of agreement. But the kind and type of evidence, those all also differ substantially. So I mean, I think that it is definitely valuable to try to give drug developers one target to look at as much as we can, as far as evidence goes, as far as biomarkers go, and we definitely do that. We are in communication with the EMA. So it's definitely a worthwhile goal, but everybody should understand that it is a different regulatory system.
Any other questions? Okay.
Hi. I'm pretty new to this community, so the first question is for the audience. I would like to know how many people here know any relative or directly purchases drugs from abroad. Please raise your hand.
I don't know what the question. Could you repeat the question?
I would like to know how many people from this audience already buy drugs from abroad, or they know any relatives that buy drugs from abroad.
Okay, so deflazacort in general is bought from abroad in the U.S. I don't know if so should we rule out deflazacort and say who else buys other drugs from abroad?
Okay, so yeah, that was basically, I just wanted to know. And regarding the harmonization with the FDA, is there any kind of, how often do you get in touch with them? Is there any way that there will be in the future some kind of a review, a statistical review as to how the drugs work over there and how this could actually be implemented over here? Because there seems to be a disconnect.
Well, I mean, again, I kind of hesitate to talk about the EMA regulations, and there's no time anyway, but they're different, and I think that's what's responsible for Andy's telling me that we're out of time, but there's so maybe. But we talk to the EMA all the time. We're on first name basis with the people at the EMA. I think I could reassure you of that.
All right. I'm sorry, Bob. We have to stop, or we'll be here until midnight. So, Dr. Farkas, thank you so very much.